All right, so if you remember, one of the things we talked about with Mendel's discoveries is the concept that we carry two of every gene and we only hand down one. And he determined that that separation, or as he coined it, the law of segregation, is basically that the copies of genes, the two copies that we carry of every gene, are separated when we make our gametes, which are our sex cells. So genes are really stretches of DNA on our chromosomes. So he didn't know that at that time, Mendel didn't. But genes are something physical in that they are found in our DNA. They're stretches of A's, T's, G's, and C's, and they're found scattered amongst our chromosomes. So, for instance, that little segment right there could be one little chunk of a chromosome, could be a gene for something, hair color, eye color, who knows. And again, here's a picture of our 46 chromosomes, at least if you are a female. All right, so like for instance, that little segment there could be a gene. All right, and if we remember, we carry two of every gene. So if that would be a segment, then there would also be a copy right here. All right, there's a second copy. And the way this works out is that one copy of the gene is on a chromosome we inherit from dad. So for instance, perhaps that one. I mean, you can't tell by looking at it, but um, let's just assume for now we inherit that chromosome with that copy of that gene from dad, and then the other chromosome with the other copy of the gene we inherit from mom. All right, and now keep in mind there are many, many genes on each of these chromosomes. And for every gene we find here, we're going to find the same gene on that partner, that other chromosome that we inherited from the other parent. So, as we mentioned, when an organism produces its gametes, or if you will, the sex cells, the chromosomes carrying the two copies of every gene are segregated. So, for instance, if we were just to focus on this one pair of chromosomes, when a male produces, for instance, sperm cells, one sperm cell is going to inherit one of the chromosomes, and another sperm cell would inherit the other, in that one of those sperm cells is going to carry that copy, that chromosome on to the next generation. The other probably won't. So each species has a specific number of chromosomes in each cell. So for instance, we as humans, our number is 46. All right, now we're not the only species with 46. And yes, there are some humans who might have slightly more, slightly less, and usually those are associated with certain disabilities. But give or take, the average human has 46 chromosomes. Most humans do. Take a shot in the dark here. How many chromosomes do you think this bottlenose dolphin has? 44. All right. How many do you think this head of lettuce has? 18. All right. They are, by the way, they are a living thing. I want to clarify that. I've been asked that before, and they are a plant, and they are indeed living, and they do indeed have chromosomes in their cells. Guinea pig. So think about it. Humans 46, dolphins 44, lettuce 18. These guys maybe fall in the middle, right? No, they have 64. So the number of chromosomes tells you very little to nothing about the species. So it says nothing about intelligence or size or anything for that matter. It's kind of random how that number evolved, so to say. All right. Now, in each of those cases, half of those came from mom. That should say half. I don't know why it says fourth. Half of those came from mom, all right, and half of the chromosomes come from dad. So, for instance, if these are our 46 chromosomes, 23 come from mom, 23 come from dad. And each chromosome from mom has a match that, in that it has all the same genes to a chromosome that comes from dad. Okay, so it's not random whatsoever. It's very, very well matched out, if you will. And those chromosomes, those matching chromosomes that have the same genes, not identical, mind you, but have the same genes, are considered homologous chromosomes. We've seen this prefix before. Homo means same. All right. So they're chromosomes that are in some way, shape, or form the same. All right. Now, for instance, if we were to look at these two chromosomes, very long chromosomes here, all the same genes would be there. Now, there's genes that affect things that are quite physical about us, hair color, eye color. So let's say hypothetically, there's an eye color gene here. Well, in the exact same spot on the other chromosome, you'd also find an eye color gene. Now, the question is, are they the same allele? May or may not be. If we looked at these two chromosomes that we already have, all the same genes, gene for gene, everything would be there. There's probably hundreds of genes on each of those chromosomes. Look down here. These guys have the same genes. All right. One of those chromosomes came from mom, one came from dad, and so on and so forth for the first 44 of the 46 chromosomes. When we get down to these guys down here, the X's, and there is a Y that we'll talk about eventually, the rules don't necessarily apply. They do, but and they don't. So we'll talk about that eventually. Stay tuned. So most cells have both sets of homologous chromosomes in our bodies, from skin to muscle, you name it. All right, and that they have 46 chromosomes. We call those cells diploid cells. Gametes, on the other hand, the very few cells within our bodies that are gametes, again, if you're male, it'll be a sperm cell, female will be an egg cell. Each of those would only have one of each chromosome. So, for instance, if we look at this picture here, the diploids have the 46 chromosomes, the 23 pairs. Gametes, for instance, like this one here, would only have one of every chromosome. And it's not a random split, it's one of every particular chromosome. If you notice, in each pair, one went missing. All right, and we call those haploids. It's almost like they're saying they're half of a diploid, but they're haploid cells. And haploids, again, only have one of every gene too. So if they have one of every homologous chromosome, they're naturally going to have one of every gene. But a haploid is a gamete. So these cells are almost always made to carry half of the genetic information to the next generation. And then in doing so, we assign each species an N number based on the number of chromosomes. So for instance, ours is 23. And I hope that makes sense because we have 46 chromosomes. Our diploids, a diploid is considered 2N, would have 2 times that number, 2 times 23, 46. So for instance, this diploid here has 46 chromosomes, 2 times the N number. Haploids are 1 times the N number, 1 times 23. All right? So if you understand the N number, you can determine the number of chromosomes in diploids and haploids from just that number alone. So let's go back to our different species here. Oops, sorry. So we have the bottlenose dolphin, and we have the lettuce, and we have the guinea pig. So remember, these are the number of chromosomes in most of the cells, 44, okay, 18, and 64. So the question is, what's the N number? N number for bottlenose dolphins is 22. All right, and then the N number for iceberg lettuce is 9, and the N number for guinea pigs is 32. Now notice, by the way, that the number of chromosomes in cells, albeit these are only 3 and include us 2 being 4, is always an even number, and that makes sense because our diploids have pairs. All right, so 2, couples of chromosomes. So that's always going to make it an even number. The N number doesn't have to be even, but the total number of chromosomes in a diploid has to be an even number. So what we're ultimately leading to is meiosis, which is a process in which haploids are made from diploids. Or in other words, we take our cells with the complete number of chromosomes and create cells that have half that number. All right? So in other words, we're going to take, for instance, the number of chromosomes and cut it in half. So if we're humans, we're taking a cell with 46 and we're creating cells that have 23. And we're not just creating half cells or half the number of chromosomes, we're more specifically separating those homologous chromosomes. So if we were to simplify a cell here, one would, let's say, 6, but 3 pairs of homologous chromosomes, we're making sure that each of the new cells has 1 of every chromosome. So if we were to think of these as 2 longs, 2 medium, 2 short, each of the haploids is going to get 1 long, 1 medium, and 1 short. All right. So and it is a 2-part process. So meiosis is one, or if you will, the first half of it, is a separation of the homologous chromosomes, or separating the pairs. So I have my little picture here, maroon and white, if you will. We're going to pretend that these guys are homologous. But one of the clever things that happens is just prior to that, the chromosomes do replicate. All right, so we create whatever, if you remember, what are called chromatids. They're attached to the centromere, much like we see in mitosis. So the chromosomes are going to replicate. All right, so albeit this picture is not as good, but it does show that the chromosomes do replicate. So to clarify what we're looking at here, these chromatids are identical. All right, so that, for instance, long chromosome is attached to an identical copy. All right, as much just like these 2 maroons are identical to each other, but don't confuse that with the concept of being homologous. So these guys are still homologous to these. Doesn't mean they're identical. It means they have the same genes. So just like the maroon is going to be still homologous to the white, having all the same genes, not necessarily the same alleles. So this pair of replicated homologous chromosomes is called a tetrid. tet is often used in reference to there being 4 of something. All right, so we actually call this a tetrid. It's a replicated pair. And one of the clever things that happens to tetrads is they go through this process called crossing over, which basically is when segments of DNA are exchanged between homologous chromosomes. So a little bit of maroon is going to exchange with a little bit of white. All right, and ultimately why that's important is because only one of these chromosomes is going to make it to the next generation, and you now have 4 unique chromosomes. You have one that's all maroon, one that's mostly maroon, a little bit of white, one that's mostly white, a little bit of maroon, and one that's all white. One of those guys is going to make it to the next generation, so you're left with 4 possible unique chromosomes, albeit very similar, but unique nonetheless. So they are now completely unique, and the thing is, every time meiosis happens, crossing over is in a different place. So let's say the next time it happens, the crossing over will occur here, but still you're creating 4 unique chromosomes. All right, so let's say hypothetically meiosis 1 ends. We separate the homologous chromosomes, which leads us to meiosis 2, which is the separation of the identical, and I'll say that with an asterisk because they're no longer identical. Crossing over truly makes them not identical, but theoretically they're attached and they're chromatids. So we separate those attached chromosomes and we create gametes that have a unique genetic complement in each. All right, just looking at this one pair of homologous chromosomes, we have 4 gametes that has, as I mentioned before, one that's all maroon, one that's mostly maroon, a little bit of white, one that's mostly white, a little bit of maroon, and one that's all white. And again, only one of those guys is going to make its way to the next generation. All right, so quick summary. Meiosis 1, here's a picture from your book. Notice how you either have the chromosomes replicating and then crossing over. Notice how they're kind of swapped between the red and the yellow from the green and the blue, those being the homologous pairs, and that's what we're separating in meiosis 1, the pairs. All right, so for instance, we have the crossing over, we have the replication, the crossing over, and then we have the ultimate separation. All right, and then in meiosis 2, we separate those attached chromatids. So now that they've been separated, we separate the attached chromatids, as we can see here. All right, the red is separated from the mostly red, the green is separated from the mostly green, and so on and so forth. And just like we see here, we're separating the attached chromosomes, creating in the end 4 unique gametes. That's important about meiosis. So we're creating 4 unique gametes. So looking at it at beginning to end, there it is, the first division separating the homologous chromosomes. There it is, the second division separating the attached chromosomes. All right, or if we look at it with our little simple case here, all right, meiosis 1, you separate the homologous chromosomes. Meiosis 2, you separate the attached. And if we look at, the original cell that would have existed, notice how this cell compares to the final cells. It has half the number of chromosomes, the final ones do, and we've separated the homologous chromosomes in each, and crossing over also occurred somewhere in between.